Hangout is on the air live. Let's see. Hey. Good morning, Scott. Good morning. How's everything going? It's good. How are you? Good. Just to let you know, I'm going to back out a few minutes early to go teach class, so don't take offense. We'll see. Just starting like Bigfoot. Yeah, I was like snorting big loud. Why is it? Yeah, exactly. So, well, thanks for coming. Yeah. Is that what it is? It's so nice. Yeah. And this is my sister Jennifer. Hi. Hi, Jennifer. It's a little greasy. You want to see someone else? I can stash them in Randy's office. So this seems to be working out pretty well. Hopefully that works. Can you check? Yeah, it's really good. Thanks for bringing me, Anna. Where are you going? Wait, here's the book. Here's the ball, yeah. You went before, like, the Senate Science Committee. Yeah, that was great. I used to go double. Now I'm single. It's hard to tell when you're joking. Six is definitely. Oh, yeah. What's that? I don't think that yeah. So, your postdoc doesn't start for a while. Okay. Is there a reason why? One. That's pretty heavy. You don't want to be there when it's setting up necessarily. That's you won't be able to do anything. So kind of thanks. Yeah, DC nice. Oh, this Mark.
Big cookies from the house.
Yeah, so sick of Maguffin. I know, I'm gonna watch it. Okay.
First mess.
Very well. Very good.
I had to go back.
Yeah.
Yeah, got it. I did see this letter was important, so I was reading the file. And then this is, I said, where is that? Yeah. What is it? So I think this out. Oh, good. Yeah, we don't do these things so often that we remember all the things. Yeah. Hey, Scott. So Jim said he's going to be like a few minutes late. So, okay. And to wait for him. Okay. Yeah, it's fine. Oh, yeah. And then it was like really very oh, yeah, that's great. Yeah. I've been practicing my signature. I asked where these are made with it. You can like this exercise. I don't know. It's just I didn't want to see myself talking all the
Good morning, everyone. Thanks for bringing this lovely weather. And I'm talking to the guests there from Scott's family. So welcome to Eugene. It's very nice to see you here. Give support to your brother. So it's very, very nice to see you. Thank you. So welcome and thank you to all of you who come to Scott's PhD defense presentation. 9 o'clock in the morning, not always easy. So thank you very much. Scott came to us from New York, so I have a brief, if you don't mind, I'll read to you. So in 2004, Scott got his bachelor's degree from GAESEO. You know what that is. And then in 2007, he started he got his master's degree. Oh, you started, or you finished?
I finished.
I finished, thank you. So he finished his, got a master's degree in hydrogeology from Illinois State University. And then in 2010, he joined us here. So last night, I was thinking, what should I say? I think the most important thing that I get a summary for the so many years Scott have been here, that he is the main driving force behind my arsenic project, and he bring the project to a very successful end. So thank you, Scott, from me.
Thank you.
And then, in addition to, so you will hear his presentations. He had made a significant contribution to the understanding of arsenic. He had one publication, and he will, and you will see here, 3 more to come. And beyond science and work, I think Scott is also a very fun person to be around, as many of you have seen that. I personally, I don't like people that much. Seriously, I don't interact with people that much at all. It's my personality. But we still managed to ride a bike from Seattle to Portland in 2 days. So that's quite amazing. So I'll always remember that. So these are a few highlights this is really a big person. And I'm very proud to say that today is the last day that he will have a chance to show you what he has done. In the near future, he will teach the summer camp here, and then he has a 3 months short fellowship job on the science policy in Washington, D.C. And then he will start 2 years, and he has a postdoc. And now, University continue his work on arsenic. So, thank you for listening to me, and Scott, please take this.
Thank you for that introduction, Shen. That's great. And thanks for all of you for being here today. It really means a lot. I know it's early on a Friday, but it's still kind of cool right now. I'm really excited to talk to you about my research today. The title of my dissertation presentation is a biogeochemical study of groundwater arsenic contamination in the Southern Willamette Basin, Oregon, USA. So I'm pretty excited about presenting this research and putting this talk together. And in doing so, I was kind of reminded and rediscovered the tragic motivation behind my research, which is arsenic contamination, which is truly a global crisis. The EPA limits arsenic contamination in drinking water to 10 micrograms per liter, or 10 ppb, parts per billion. Yet many wells in the United States exceed these drinking water limits, and it's a health risk and a health problem for many people in the United States. It's an even bigger problem globally. 100 million people worldwide suffer or are exposed to arsenic-contaminated groundwater that exceed these levels. The World Health Organization called it, in their latest report, the largest mass poisoning in history. Specifically referring to just the area in Southeast Asia, Vietnam, Cambodia, Bangladesh, where tens of millions of people are consuming arsenic-contaminated groundwater every single day. Exposure to arsenic in the long term is an especially gruesome way to suffer. It starts shutting down some fundamental processes that your body needs to do, such as within your skin, regeneration. You start to develop lesions on your hands and your feet. Your organs can begin to shut down completely, and the litany of different cancers can develop. So this is a serious problem, and again, it's a global crisis. So that is the motivation for understanding the cycling and cause of high arsenic concentrations in groundwater. The difficulty is it's not well understood. What are the mechanisms that elevate groundwater arsenic? And what are the biogeochemical processes that pull it out of groundwater? So let's begin where arsenic begins. In the Earth's crust, arsenic is the 20th most abundant element. It has an average of 2 milligrams of arsenic per kilogram of rock. It's a decent amount of arsenic. And arsenopyrite and orpiment are the most abundant sources of arsenic in the Earth's crust. These minerals are relatively stable under anaerobic conditions, but if we expose them to oxygen on the Earth's surface, they begin to break down, and they can be a significant source of arsenic in the environment. Ferrihydrite, although ferrihydrite doesn't have arsenic incorporated into its structure, arsenic is often a contaminant within the mineral itself. So ferrihydrite, being a very abundant mineral in sedimentary rocks, can also be a significant source of arsenic in the environment. Igneous rocks like andesite have arsenic concentrations about 1 to 5 milligrams per kilogram. Basalt can be even higher. Even some basalt can be up to 100 milligrams per kilogram. And this is notable because a lot of the current locations where there's arsenic contamination are proximal or near igneous mountain ranges where sediment is sloughed off, transported, eroded, and deposited. So this is often a source of arsenic in sedimentary units. So this is not really a problem as far as human health goes, because most people aren't eating orpiment or arsenopyrite. But the problem arises when there's a transition from the solid phase to the aqueous phase, where it then enters the human exposure pathway, such as groundwater, right? People are extracting groundwater to drink it. Often, that's the only potable source is extracting it from the ground. The two most common species are arsenate down here. That's AS5, that's the oxidation state. It's the oxidized version, oxidized species of arsenic, and arsenite, arsenic 3. It's the reduced form of arsenic. These account for about 90 to sometimes 100% of the arsenic that you encounter or see in groundwater. And it occurs from anywhere from 0, it's not present, to 1 part per million concentration. The toxicity of these two species is significant. They are very toxic. And arsenite is considered more toxic, and it's more soluble. It's more soluble in most groundwater situations. Then there's organic arsenic, which is much less abundant than these inorganic arsenic species. There's monomethyl arsenic acid, I'll refer to that as MMA, and dimethyl arsenic acid, I'll refer to that as DMA. These are organic species. You can see the monomethyl arsenic acid has 1 methyl group, and the dimethyl has 2 methyl groups. What's happening here? And they occur anywhere from 0 to 10 parts per billion, ppb. They are toxic to humans, but their relative toxicity to arsenite and arsenate and to each other is not widely agreed upon. So what my research is focusing on is the interaction between the solid phase and these species here, and how they cycle back and forth within groundwater situations, groundwater environments. So arsenic is cycled in many different reservoirs. This is a spatial view of arsenic cycling and the flux between different reservoirs. The red lines indicate a relative abundance of arsenic flux between those reservoirs. Notice how thin the groundwater red line is. This is a reflection of the relative insignificance within this conceptual model of arsenic flux within groundwater. This model indicates that groundwater flux of arsenic into the soil, into the atmosphere, rivers, lakes, and streams is not necessarily important. Compare that to the soil, the flux represented in this conceptual model. It's a much more significant amount of arsenic being transported from soil into groundwater, into lakes and streams, and into the atmosphere, as well as agriculture, rice paddies, for example. This is the current understanding of flux within the global arsenic cycle. Let's look a little more comprehensively at the soil arsenic cycle. So this is a conceptual model, modern conceptual model of arsenic cycling in soil and surface waters. It is comprehensive. Notice that arsenate and arsenite are included, and the reduction and oxidation reactions between these two species. That arsenite can be methylated to MMA and further methylated to DMA. There also can be demethylation pathways where you remove those methyl groups, and it goes back to arsenite and arsenate. This model also considers the interaction between all of these aqueous species and the solid phase. Maybe there's precipitation or adsorption or dissolution of minerals. This is a comprehensive conceptual model of surface arsenic cycling for soil and ground and surface waters. Compare this to a notable science paper in 2003 for the conceptual model of groundwater cycling. There's arsenate as a source from these iron and arsenic sulfide minerals being cycled through and transforming to arsenite at the expense of some organic material here and producing CO2. And then there's some interactions between arsenic 5 and iron and aluminum. Notice the absence of organic arsenic pathways of oxidation and interactions between the solid phase and dissolution and precipitation reactions. Importantly, there is no organic arsenic. This paper goes on to say that it is apparent that natural organoarsenicals do not contribute substantially to the biogeochemical cycle. This is still the current conceptual model of groundwater arsenic cycle. So let's take a more comprehensive look at groundwater arsenic cycle and how arsenic can actually enter the dissolve phase within groundwater systems. One way is mineral oxidation. On the left here is an example. This is an aquifer. There's a water table. If for some reason you lower the water table due to, say, extraction, or you overpump, and maybe there's a drought or something, the water table lowers, and if pyrite is within that aquifer, what was once an anaerobic environment then becomes aerobic, and you introduce oxygen to the system, you start oxidizing pyrite. In pyrite, arsenic is also often an impurity in pyrite. So the oxidation and dissolution of pyrite can introduce arsenic into that groundwater system. Another way, but similar mechanism, is through acid mine drainage. Mineral oxidation of ore is extracted mostly where the tailings are piled up next to a mine, and it's essentially like you're concentrating pyrite and oxidizing that really quickly because it's not stable in an oxygen-rich atmosphere like our own. So that'll produce sulfuric acid and then often bleach a lot of arsenic into the soil, but also get into the groundwater and subsurface. Another very important way that arsenic can enter the dissolved phase of groundwater or be removed is through something called sorption. Sorption is the adhesion of an atom, an ion, or a molecule onto the surface of a mineral due to a charge difference. And this cartoon here, this illustration on the left, indicates two different ways. An arsenate molecule can adsorb or sorb onto ferrihydrite. So there's empirical evidence to support this process and its relative significance with respect to pH. So if here is a measure of arsenite and arsenate, and on the y-axis and on the x-axis is pH. As you can see, you probably figured out already, that pH is a significant control on the amount of arsenic that sorbs onto these mineral surfaces. More so for arsenate than arsenite. Another important sorption is on iron sulfides here and here. Just I want the take-home message from this figure is that, again, the sorption of arsenite is very strong on iron sulfide species, but it varies with each. The last part of my introduction, I'll discuss the last control of groundwater arsenic is microbial transformation. By microbial transformation, of course, we're talking about microscopic organisms within the aquifer. These microorganisms can be, these communities of microorganisms can be very diverse. And when there's diverse microorganisms, you have diverse metabolisms. So diverse metabolisms mean that each microorganism is going to utilize organic material for energy in different ways. So, for example, in this cartoon here, arsenate reduction is a primary control over the concentration of dissolved arsenic in groundwater. So an arsenic-reducing bacterium, represented by this green oval here, will take arsenate in the oxidized form, and if it finds some organic matter it wants to oxidize and you can capture energy from, it's going to transfer the electron from that organic matter. To capture energy, it's got to move that electron somewhere. And it'll do that by moving electrons to the arsenic, which reduces it to arsenite. So by microorganisms consuming organic material, it's changing the speciation of arsenic species from arsenate to arsenite. And because arsenite is more likely to exist in the dissolved phase, it's directly changing groundwater arsenic concentrations. If oxygen enters the atmosphere, this arsenite could then oxidize and resorb as arsenate after it's oxidized. So these are primary controls that microorganisms have over arsenic concentration. There are secondary controls that microorganisms have over the groundwater arsenic concentrations. The first one here, example, is mineral reduction and dissolution. One example of that is if you have an iron-reducing bacterium that, again, it's looking to capture energy from organic material. So it takes that electron and transfers it from ferric iron, which is insoluble with ferric iron minerals, and reduces that ferric iron to ferrous iron, Fe2. And ferrous iron is very soluble. So in doing that, it's reducing the ferric iron to ferrous iron and dissolving that mineral. But that has a direct link to arsenic concentration because of the sorbing locations of arsenate onto ferrihydrite and other ferric iron minerals. So once you dissolve those minerals, you're taking away those sorbing locations and then releasing arsenic into the ground. And then lastly is sulfate. Sulfate reducing bacteria will reduce sulfate, produce sulfide. And that sulfide, if there's also iron reduction happening at the same time, it will combine to create an iron sulfide. And those iron sulfides can pull out groundwater arsenic. Here's an experiment by Southfield and Bostic, 2009. Basically, they had 5 samples of ferrihydrite-rich aquifer media. They added varying amounts of sulfate, and this was also contaminated with arsenic in the experiments. They added a sulfate-reducing bacterium, E. vulgaris, and what they saw is that there was sulfate reduction, which combined with ferrous iron, precipitated iron sulfide, and you had a reduction in groundwater arsenic, or in the experiments, over time. And the color changes this iron sulfide precipitate. So the more precipitate they observed, the more arsenic was removed. Okay, so that is the background. My research, overall research question is, how do microorganisms affect groundwater arsenic? And I'm going to, for this talk, I'm going to direct the rest of this talk to answer 2 questions, one of which is, how does arsenic cycling operate within groundwater? How does it cycle between the mineral phase and the dissolved phase? And second, is bioremediation a viable strategy for combating the global arsenic crisis? If it is, then we can help a lot of people by a better understanding groundwater arsenic contamination. So in order to address these 2 questions, this is my study area. I utilize the high arsenic contamination within the Fisher Formation. So Eugene's just north of here. This area is just south. This is the Fisher Formation in red. This picture is taken of this area from Mount Pisgah, which is pretty much up there in the corner. And Cresswell, a friendly town, is right over here. Recharge in this area is in this fish formations over here, and groundwater flow is to the northeast. High arsenic concentrations are in the anaerobic covered area, confined area. The fish formation is a volcanoclastic sandstone, tuff and ash. It's overlaid by these igneous deposits and river sediments, which creates that anaerobic reducing aquifer. And again, just like the examples I gave in the introduction, the fish formation, a lot of that arsenic is probably coming from the Dakota Cascades as it was being eroded and transported down, deposited as a marcosic sandstone and a volcanoclastic sandstone. And the source of that arsenic was probably salt and andesite. So I had 2 approaches, 2 experimental approaches. The first one was laboratory microcosm experiments. There's Jordan over there, he's in the back. He was a big help in these laboratory experiments. In those experiments, I simulated aquifer biogeochemistry under controlled conditions. And then in the field-based aquifer tests, which were in situ tests, explored in situ biogeochemical reactions using a tracer-based solution method where and monitored over time. So I'll talk about the lab experiment first. This is Jordan and I collecting aquifer sediment from a well outside of Cresswell. The idea here is that we would take aquifer sediment from the bottom of a contaminated well. The arsenic concentrations within this area are very high, 2,000, upwards of 2,000 ppb. So many orders of magnitude greater than the maximum contamination limits. As we collected these sediments, we were also collecting the indigenous microbial communities that existed within this aquifer that we were hypothesizing are responsible for the arsenic contamination. Then, using that, those sediments, we built a series of 4 reactors, putting them in 1-liter bottles. We built a synthetic groundwater based on the chemistry of the aquifer. But we added, instead of arsenite, which is the contamination in the aquifer itself, we added arsenate so we can witness those redox reactions, if any. The first reactor, A, we killed because we wanted to make sure that any other reactions that we witnessed that were different than A, we can attribute to a biological process. In reactor N, we didn't add any ethanol, we just added a little sulfate to see what would happen. So therefore, in reactor E and ES, we could attribute differences between those and N to the ethanol addition. In reactor E, we added ethanol but no sulfate. In reactor ES, we added both ethanol and sulfate, ES. What were the results? Well, we incubated them for 2 months in the dark at 16 degrees C. So the results here, this is iron, ferrous iron, this is the reduced form over time, and we see iron reduction beginning. Here's your key for this part of the talk. I'll talk exclusively about E and ES, but I'm incorporating A and N into many of these plots to assure you that they're a biological process and an ethanol-driven process is at play. E is the open circle, ES is the closed circle, and that indicates that there's an additional sulfate in these experiments. So what do we see with iron reduction? Iron reduction begins within the first week. We see more iron reduction happening when there isn't sulfate than when there is. This is solid phase analyses from the beginning of the experiment, and then these are solid phase analyses of the end. These data agree because there's more ferric iron reduction happening in ES, which produced more ferrous iron. We also wanted to get a handle on the timing of ferric iron reduction. So using fluoride, coincidentally, is a really useful tool because fluoride absorbs onto ferrihydride. And I included chloride up here because it's a conservative ion. It is non-reactive, so if anything changes here dramatically, we can subtract that from the fluoride. But fluoride is consistent. In the fluoride, and you see a desorption from perihydride into dissolved phase, which stops around day 17 for both E and ES. And there's more fluoride in E, which agrees with these data here. And it puts a nice window on our ferric iron reduction. So here's that iron reduction data here. Note that pH has a significant decrease over time. It is decreasing dramatically from about 7.8 to about 6.1 by day 12 in the ethanol-stimulated experiments. This is because our ethanol here is being oxidized, and as it's oxidized, it produces acetate, produces H2, which is fueling a lot of the other reactions here, and it produces a proton. The proton, or H+, drives down pH. Notably, sulfate here, we didn't add any sulfate in E, so it's pretty low, but ES has the sulfate concentrations we added, and it depletes over time. It's gone by day 32. We also see the sulfate reduction here in sulfide concentrations. And in fact, all biologically active reactors have sulfide, meaning even the ones that we didn't add sulfate to or ethanol are kind of churning out some sulfate reduction. It's important. So, what's he saying? So what does that mean for arsenic? This is the dissolved arsenic in both arsenate and arsenite. Here on the bottom here is arsenite. That's the reduced form, arsenate. And this is the total dissolved arsenic detected in these samples over time. Well, in reactor E and reactor ES, we see immediate absorption of all the arsenate. Then it begins to be reduced in both samples, in both reactors. And so you have an increase in arsenite here. But then this is where the two E and ES split. They do something very different. In E, where there's no sulfate addition, it increases and then just gradually gets higher and higher arsenic concentrations. This is also visible here in E, in the total dissolved arsenic. E just gradually gets higher. However, in ES, something unusual happens. You have the initial arsenite reduction, arsenite production through arsenate reduction, and then all the arsenic drops out. A significant amount of arsenic drops out of the dissolved phase from all the experiments. There is standard deviation on here for all four bottles. So we saw that happen in all 4 bottles. So that was really interesting to see. But then the arsenic carries on and eventually increases. The end result of these experiments is that both E and ES have pretty much the same amount of dissolved arsenic at the end of the experiment. So In the solid phase, we see an iron sulfide precipitate in reactor ES, which is expected. We added lots of sulfate, we added ethanol, so sulfate is being reduced. And we don't see any iron sulfide precipitate in reactors N or E or in the bedrock. So we were thinking that maybe the arsenic was being sequestered with that iron sulfide. And maybe it was, but this is the before and after of the experiment. In the solid phase analysis of arsenic in iron sulfide, there is no significant accumulation of arsenic within the iron sulfide as an impurity. So that was one idea, that as iron sulfide precipitates, it's going to incorporate and pull out arsenic within the mineral as an impurity. That didn't happen here. In fact, E has a little bit more than ES. So what we do see is an increase, significant increase in adsorption of arsenic and an increase in absorption of Fe2. So putting all of this together, we don't see any significant sequestration of arsenic that persisted during the experiment. We do see a significant amount being pulled out just for that one sampling event. So this is a plot you're going to see a few times for the rest of the talk. And it's, I'll just explain it now. It's always going to be total arsenic concentrations on the bottom, a long time. And then this is a geochemical, qualitative, somewhat quantitative illustration of what's going on. So the thickness of the line will indicate the relative abundance of the process that's happening. And then the length this way will indicate the timing of it. And so in reactor E, it's pretty simple. We see iron reduction happening. Really, very little sulfate reduction. We don't see any iron sulfide precipitate, but arsenic reduction is happening pretty much from day 3 to about day 42. And that resulted in an increase in total arsenic. We see that sorption in the beginning, and that total. Compare that to reactor ES, where we added ethanol and sulfate. It gets a little weird. We have the absorption of arsenate being reduced to arsenite, then that drop, and then the overall reduction of arsenate to increase those concentrations. What I think is going on here is that Fe reduction happened and sulfate reduction occurred, as well as arsenate reduction. Through the iron reduction and sulfate reduction, we created this window where there's an iron sulfide precipitate. Did some saturation calculations. It was saturated at this point, so it's dropping out iron sulfide, but it didn't incorporate it as a impurity within that mineral. But iron sulfide is a really strong adsorbent of arsenite. So what we think is happening here is that arsenite adsorbed onto the iron sulfide precipitate, which we observed in ES solid phase analysis, and then desorbed due to pH controls. In conclusion, the sulfate ethanol decreased iron reduction and provided more adsorption locations for iron oxyhydroxides. Sulfate ethanol addition produced more iron sulfides, and then that provided additional adsorbing locations, but ultimately the pH controls influenced the adsorption and then desorption of dissolved arsenic between the two phases. So, go ahead.
Yeah. Is arsenite that's in the adsorbent experiments?
They're probably both absorbing, but arsenite absorbs more strongly onto the iron sulfides. And that's the reduced form that's more likely to go into the dissolved phase.
But the one that's sensitive to pH. Yeah.
They're both sensitive to pH. Arsenate is more sensitive to pH for ferrihydrite. Arsenite is more sensitive to pH for iron sulfides. Yeah. Good question. So with the field push-pull tests, this is the field in situ experiment. We injected these controlled solutions into well 13 here, where there is about a 2,000 ppb arsenic contamination problem. So what is a field push-pull test? This is the cartoon. There's two of them. The first one was a control test. We added sulfate and arsenate into the solution and injected it. This solution also simulated a lot of the aquifer chemistry, as well as contained a tracer. And the second test is we added ethanol, sulfate, and arsenate to see what would happen when those microorganisms within the aquifer were stimulated with ethanol. So the solution, you push it into the ground, into this isolated part of the aquifer, and then over time, it starts mixing with groundwater, fading, and you're sampling the whole time. So, and you can monitor the amount of mixing that your solution has been doing with groundwater based on that conservative tracing. So let's just jump right into the arsenic data. In the control test, there's not much absorption that happens at all with arsenate. What happens is arsenate drops and arsenite is produced almost immediately. So you have onset of arsenic reduction very, very quickly. This produces arsenate a little bit, and arsenate starts to desorb here, but then it drops considerably, and arsenite is produced, and they kind of plateau. It's a simple arsenate, a little bit is being reduced, desorbs, and then it produces a whole lot of arsenite, and they both are stable from about halfway to the experiment to the end. Oh, and I should say that these dashed lines are the mixing controls. So for the rest of the plots that you'll see, these data here represent what the concentrations of whatever species they're talking about would be if it was just groundwater mixing. So no biological, no geochemical reactions, just if you mix the solution to the groundwater. So we calculated that through background concentrations and the conservative tracing. In the ethanol stimulated test, it's very interesting. The arsenate here drops down and is at very, very low concentrations. You have a little peak in the middle here, and then it's at very low concentrations again. Arsenite, on the other hand, jumps up because it's being reduced, arsenate's being reduced, producing arsenite, but then that also drops down significantly. Then increases again, and then drops down and maintains fairly low levels, much lower than you would expect just based on mixing. So a lot of arsenic is being pulled out of the dissolved phase here. So what's going on? Why is that happening? Well, if you look at sulfate reduction, the control test is the circle, the ethanol test is the square here. Sulfate reduction is not occurring in the control test, which is actually accumulating because more sulfate is washing in. Sulfate reduction occurs in the ethanol test about 6 or 7 days in. This is also reflected in the sulfide concentration. Sulfate reduction produces sulfide, and you see sulfide production happening until about day 32. And iron reduction, importantly, is delayed. Iron reduction doesn't begin until about day 20. Here, you have an increase in ferrous iron. It's also confirmed in some solid phase analyses of ionically bound arsenic, which is potentially in iron oxide, iron oxyhydroxides, which decreases around day 20, and then arsenic in crystalline ferric oxyhydroxides decreases around the same amount. So we have a pretty good window on sulfate reduction and iron reduction. So the iron sulfides were precipitated. All these solid phase analyses are for the ethanol test. Iron sulfide was precipitated in the last 2 sampling events. That's when it was detected. So at some point from day 23 to the end of the experiment, iron sulfide was precipitated. Arsenic in iron sulfide slightly increased over time. The data is a little more. Importantly, though, arsenic and orpiment began precipitating around day analyses where this is a relative ratios here, but arsenate decreases over time, arsenite increases, and then I've blown this relationship up. Orpiment goes from a fraction of a percent of total arsenic to 5% of total arsenic. So there's a significant precipitation of arsenic that I think is pulling out a lot of that dissolved arsenic. So this plot again, let's look at the biogeochemical networking of these tests. Total arsenic, we injected arsenate, some of it absorbs, some of it gets reduced right away. It increases in concentration, then plateaus. It's just arsenate reduction. There's no detectable iron reduction, there's no detectable sulfate reduction, there's no iron sulfide precipitate. You just have a lot of arsenic in the dissolved. Now, in the ethanol test, this is very interesting. This is total dissolved arsenic, remember, total dissolved arsenic. We see that dramatic drop. All the arsenic in the water just decreases to about 100 ppb, where it started at 2,000, then increases again, and then decreases to relatively low concentrations. A lot of arsenic is removed from the dissolved phase here. Why is that happening? Well, like I said, we have a delay in iron reduction, which I think is important. Sulfate reduction occurs earlier, and arsenate reduction happens almost instantly. Arsenate reduction happens within the second sampling event. So the combination of sulfate reduction and arsenate reduction with the delayed iron reduction allows this window for orpiment saturation and sequestration of arsenic here. So orpiment begins to precipitate. We saw that in the solid phase analyses as well, and pulled out arsenic. This lowered the arsenic concentrations, total arsenic concentrations, and maybe some of it was incorporated into the iron sulfide as well. So this is a 75% reduction in total dissolved arsenic, as is this area here. However, I'll get to this in just a moment. So conclusions for the field test are, we saw a 75% reduction of dissolved arsenic. We saw that twice. We saw an effective sequestration of arsenic by orpiment precipitation. And iron sulfide also sequestered some arsenic. So what about that other removal of arsenic from the dissolved phase? Well, can biomethylation account for this? I mentioned earlier that about biomethylation. This third part of my talk will be about this. So the problem here is that these data do not explain these data. There's no precipitation of orpiment here. There's no remarkable adsorption that's happening, and there's no inclusion of arsenic in the iron sulfide. So can biomethylation account for this? I spoke earlier in the introduction about biomethylation. You have these two species, arsenate and arsenite. There's an oxidation and a reduction reaction happening. These reactions are fundamentally important to capturing energy for arsenic-reducing microorganisms, meaning that there's organic matter, they want to collect the energy for growth, reproduction, cell metabolism, cell maintenance, all that good stuff, from organic matter. In order to do so, they need to transfer electrons to capture that energy. It's an energy capture system. These are those sort of reactions. However, MMA and DMA, the methylation of arsenite, adding these methyl groups is not an energy capturing process. It's a detoxifying process. And in fact, microorganisms will add a third methyl group, trimethyl arsenic oxide. So TMA. This detoxification method is thought to be a detoxifying method by increasing volatility. These microorganisms are adding a methyl group here, then adding a second one here, and adding a third one. So the idea is that when arsenic gets too high in these environments, they'll start and switch on this gene that starts adding these methyl groups, not to capture energy, not to grow and reproduce, but to detoxify their environment. So they add these methyl groups, they increase the volatility, and then these arsenic species, through different chemical gradients, will diffuse and move away. Last year, Shuchen and I and our co-authors put out a paper looking at the significance of organic arsenic Argentina and arsenic, methylated arsenic, and inorganic arsenic that is consistently between a fraction of a percent and 10%. At most, you'll see about 10% of methylated arsenic of the total dissolved arsenic. And so, is this a significant process in arsenic cycling? Can that volatilization, detoxification of arsenic be a meaningful part of arsenic cycling within the aquifer? Well, we found that of the biomethylated arsenic, 0.4% was 0.4% of total arsenic. And the DMA was produced at 2 ppb per day. 0.09% of the total dissolved arsenic per day. So that might not sound like a lot, but if you're producing 1/10% of all the arsenic into this, and you're just kind of moving it through this conveyor belt of methylation every single day, that actually is a significant amount of arsenic cycling within the organic methylating cycle. So here's the organic methylated arsenic data for the ethanol injection test. So as you saw the arsenate and arsenite data before, here's the MMA and DMA data. The arsenate data, as we saw, reduces really quickly because you have arsenite being produced due to arsenate reduction. But by the second sampling day, we already see MMA and we see DMA. And DMA is being produced rapidly. This is not surprising to see this difference here. MMA actually has a hard time permeating cell membranes and get stuck in cells, and then they'll get methylated again to DMA and make their way into the dissolved phase more easily. So DMA concentrations are typically high. So the way I think about this is that if the methylating begins, if these microbes start methylating, it's kind of like a conveyor belt through the methylating process. It goes from arsenate, they reduce it to arsenite. They add a methyl group to MMA, then the DMA, then it goes to TMA, and then that's how they're detoxifying the environment. So this is these are the in situ rates of organic arsenic reduction that I just mentioned here, compared to the 2.0 ppb per day and about a 10% being produced per day. In the ethanol stimulated test, we see that DMA is produced at 9 ppb per day, and nearly 2% of all the dissolved arsenic is being methylated every single day. So that is a 20-fold increase in this ratio. And if you look at DMA to the total dissolved arsenic that is in the groundwater, the DMA is shooting up, methylating, pushing up the TMA, volatilizing that arsenic, and dropping the total arsenic concentrations in the groundwater. The percent of organic arsenic here of total AS also increases during this experiment to about 14%, and it's sustained. So it's almost like they've switched on this methylating microbial process, the methylating, and for an 11-day period, they sustain this and lower these arsenic concentrations considerably. So in conclusion, biomethylation effectively removed 75% of dissolved arsenic for 11 days. The ethanol addition yielded a surprisingly fast biomethylation, 20-fold. And importantly, ethanol accelerated metabolic detoxification. Remember that those reduction and oxidation reactions are energy capture reactions. They are there to gain energy and promote growth and ATP. But that detoxification is not that. They're actually expending energy to do that. And the fact that ethanol can accelerate that process just by providing organic matter is really notable. So in review, my laboratory experiments show that the importance of pH, adsorption, and the timing of redox reactions are worth consideration when considering arsenic concentrations within the cycling of groundwater arsenic. And the field experiments demonstrate the importance of biomethylation. These results suggest that there are two promising methods for groundwater arsenic remediation. One is the precipitation of curcumin, and two is the biomethylation of arsenic and the volatilization out of the aquifer. And that biomethylation is capable of being accelerated just like an energy generating process. So I'd like to acknowledge my advisor, Yushen. Thank you very much for everything done. My committee, Mark Reed, Jim Watkins, and Scott. Scott has been especially great just providing lab time and resources for me, so I really appreciate that. Thank you. Edward and Greg, thanks because you guys were on my council committee. Don't forget that. My co-authors, Matt, Steve, Ashley, Ben, and Jin, thank you very much. Jordan, couldn't have done the lab work without anybody. Thanks. Dominic, Chris, and Kyle were also extremely helpful in the lab. The funding, NSF, thank you. The UCOR and the department actually helped me out some. The Department of Geological Sciences, great department. I couldn't ask for to be a better part of a better group of people, specifically Ray, Marley, Josh, and Becky. Thank you so much for everything you've done. I can always count on you. There's so much. And the dog staff, Dave, Sandy, Marlon, and Sherry. There's Sherry, miss her. She's listening in. And all of the hardworking undergrads that I've had the pleasure of knowing. Best of luck to you in the future. And all former and current UO dogs, faculty, graduate students, especially Al, Christina, Randy, Rob, Dan, the Rez, Berg, Will Mines, stand, rise, and add cares. T. Murphy. Yeah, sure.
Any question for Chuck?
Sure, Doug.
Yeah, that was really nice.
Thanks.
How would you bioremediate with those processes?
Sorry.
How would you do the bioremediation or the remediation of arsenic with those? How would you actually put it into practice?
So I think you'd have to understand the local geochemistry, importantly, and again, the biology is important. But if there's arsenic contamination in an aquifer, there's going to be arsenic-producing microorganisms. So it would be probable that you can do it. You'd have to control iron reduction and somehow delay it, and you promote sulfates. You add sulfate, you add some sort of amendment that promotes the metabolic detoxification process, like ethanol. And
So you have to do that in situ, like, or you, I'm trying to think how you're going to remove the arsenic from the water that people drink. You do it all by pumping things into the ground, or you do it
yeah,
after you take it out of the ground? Or?
yeah. Can you guys hear Doug's question? Okay. So that's a great question. People are remediating arsenic. There's a couple of different ways to do it. One way is to remove it, and externally you can go through a series of different processes to remove the arsenic. It's expensive, it's time-consuming, it's really hard to scale up. So with large populations, and especially dense populations, it's really difficult. But the idea here is that if you can kick in biomethylation on a large scale in situ by promoting it with sulfate reduction or just or the biomethylation to warm the precipitation would be sulfate reduction. But if you just if you don't provide the sulfate and promote the biomethylation with organic matter amendment, then that would volatilize, and the idea being that it would be removed from that specific location within the aquifer. They're migrating, they're volatile, so they would be going to other places in the aquifer, but if you focus your extraction for potable water in that area, then that might be a reasonable way to go about it. I certainly think that it needs to be more understood. This is just the first step in biomethylation. Come on.
Can you add the precipitation of open by providing irrigation sites for this?
Maybe, yeah, I think, so like if you added orpiment down, like somewhere down there and then a nucleation site for it, I don't know, maybe, yeah. And Tomas' question was, could you promote the precipitation of orpiment more if you added a nucleation site in situ? I don't know. It's an interesting thought. Mike?
Obviously, arsenic is something for that to have in drinking water. But there are other elements in the same neighborhood periodic table, selenium.
Yeah.
To what extent do those elements, bad players, track arsenic in groundwater and natural systems?
Track is in process or
Trackers are usually correlated or highly uncorrelated.
I think there's like here we don't see mercury or anything like that. But I think that typically if you have arsenic contamination, you might have other metalloids being contaminated, but for the most part it's arsenic is the problem. Then like in parts of the San Francisco Bay, there's a selenium problem. In Tennessee they have mercury. From my experience and my readings, typically there's one metalloid problem that's being created. Becky?
Kind of a general question. So it seems that many of these iron elements and all are much more soluble than the used one.
Correct.
And I mean, it's intuitive, I suppose, but I just wonder.
For arsenic,
Just in
in general, it's because of absorption. So, and it's pH control.
Also, not intuition. So tell me a little more. how that works.
So there's different charges, and let's just use arsenic and arsenite, and Fahrenheit, right, for that matter. So those are charged differently, and the charge surfaces of the minerals are will interact with Fahrenheit, right, and this arsenic species. But the charge of the solution is dependent on pH. So there's one charge in the water. And so basically that dictates how much and which of those species will adsorb onto the mineral surface. And I think that's the case with a lot of other minerals. Certainly, Matt Paul's out of stock, he's talking about manganese and adsorption properties there. Joe?
You said that one of the big sources for arsenic is the salt and andesite, and sometimes you sort of that as igneous rocks in general. Do you get a lot of arsenic in more differentiated igneous rocks, like would granite on a lot of arsenic in it?
Granite doesn't actually have that much arsenic. I'm not entirely sure why.
I'm
But
just curious why.
I don't know. Yeah, so it's more of like the intermediate mafic.
Okay, that's what I was
Yeah, so there must be some sort of fractionation process.
okay.
So what is Crestwell doing at the moment to stay happy with their groundwater?
What is Crestwell doing at the moment to
Stay happy.
stay happy?
Yeah.
Well, they're friendly, one. So what they're doing is it's actually not a big problem in Crestwell because they're on town water. So what they're extracting a lot of water, then treating. There's a huge water treatment plant, which I think actually had some controversy recently because they didn't do something right. It's escaping me at the moment. But it's a problem for the residents of Crestwell that are outside the town limits, which is a minority of the population, but there's plenty of residents in Crestwell. Sherry, how would you say, 100 people, less, are drinking out of well water or using well water?
Sorry, please hear me.
How many of the residents of Crestwell are still using well water? Hundreds?
Probably, yeah, hundreds to start with, say that. Yes. Yes.
But they're also like up on the hill, if you go up into the recharge area, arsenic contamination is not a problem. Because there's the meteoric water, it's a lot closer to the source, which brings in oxygen. And that oxygen is pretty much keeping arsenic in the arsenic form. And so since it's in the arsenic form, it's absorbing, and it's not a problem. There's really low levels of arsenic concentration. Once you get into that reducing zone, the reduction process is happening, you're desorbing a lot of arsenic and a lot of contamination. Sam?
So building on that then, if the solution as a resident in Crestwell is to get to these water sources that are more directly from the meteoric component rather than the groundwater that's been hanging around for a while, doesn't that mean that they have problems with other kinds of contamination? Because I know, I grew up in the South Sea, and if you're drawing on deep aquifers, then you're a happy person because that's the water that at least recent human activity hasn't gone and made a mess of it. But if you go up the hill and you get your sources that are more meteorically influenced, then you're drawing on whatever people have thrown fertilizer in and all that.
It's a great point, and I think that point is even more important in places other than Crestwell, like the densely populated Bangladesh or something like that, where surface water is disgusting. And your option is, do I drink this clean, arsenic-contaminated water that will kill me, but I can't smell or taste? It tastes awesome. Or do I drink this sewage-like water on the surface? So, like, it's kind of what you're getting.
You want to die, right?
Right, yeah. Pick your poison. Ben, yeah.
Back to your ethanol test, is there any threshold you see for biomethylation of arsenic? And if so, is that concentration targeted specifically to be arsenic-producing bacteria?
I don't think I understood your question.
Is there any concentration of ethanol where you don't see biomethylation?
Yeah, that's a really good point.
And is that concentration targeted to just the arsenic-producing bacteria?
Is it targeted just to what?
Just the one bacteria, like the arsenic-producing bacteria.
Well, that's a question that I can't answer because I don't have a phylogenetic catalog of the bacteria in the subsurface. So the first one, I would be hesitant to call out an actual concentration of ethanol because it's one test. But sure, we see, I mean, that graph, this graph here, where you see, maybe here, where what concentration is ethanol at this point? Because biomethylation just kind of shuts off. Right? So yeah, maybe that's it. I think that's a starting point, like at least to give you a ballpark window of like, all right, let's look at getting ethanol concentrations above this in the next study, and look at what the biomethylation rates are at that point. And then does it track? Do we see that tailing off of biomethylation at the same concentration? Yeah, that's an interesting question. Sam?
I think you might have mentioned it, but how does the biomethylation process
I haven't talked about that, and it's not really known. The biomethylation in groundwater of arsenic is not studied extensively at all. In fact, there's only 2 people studying it, including one of them. And pH controls on the metabolic process of detoxification. Is that your question? Don't know the controls on pH. We don't, so there's not a significant pH change here. But in the laboratory experiment, there is a significant pH change, and there are different methylating patterns that we see. There's that big pH drop. So maybe we can discern some sort of pattern between the changes in pH in the laboratory experiment and the metabolic processes versus this. But also then you get into a little bit of trouble because the microbial community in the laboratory experiment is almost certainly not as diverse and robust as the one that we are in situ, because you're losing a lot of those microorganisms. You don't want to, but, you know, just doing the process of having a laboratory experiment. So yeah, that's an interesting thing too. I think if you were looking at it through the lens of how do we biomethylate for remediation purposes, you would want to not expend too much of your resources in an experiment messing with pH, but maybe control for like the same pH within the aquifer. Because in that way, that's what the microbes are used to. And you start like acidifying or maybe creating an alkaline groundwater environment that might really help.
Okay, let's start again. So do we still have this one? Or
Yeah, it's ours for 57 more minutes.
Okay, all right.
But you guys can